Hello, my name is Marom Bikson, and the point of this lecture is to introduce you to how computational models, computer simulations, can be used to inform the design of brain stimulation or neuromodulation devices. And I want to specifically focus on the example of home-based devices that are used to treat chronic pain and things like migraine and neuropathic pain. So I'm going to be discussing things in terms of this particular use case. The principles I'm going to be discussing are translatable to any brain stimulation or neuromodulation device. Now, there are different types of brain stimulation or neuromodulation devices, and usually the name of the device tells you a lot about how it operates. So I've listed some here, and I actually classify these according to these broad categories. So starting first with implanted devices. So this could be something like deep brain stimulation, where an electrode is implanted deep inside the brain and energy is delivered therefore through the electrode to a deep brain target. You can see how the name tells you a lot of how it works. Same thing with spinal cord stimulation. In this case, this would be an implant with an electrode near the spinal cord. And so when energy is delivered into that electrode, it's also delivered into the spinal cord. Now, there are other classes of devices. These are non-invasive, non-implanted devices, but I'm putting them in this in-hospital category. This is because they're either so expensive or complicated to use or have certain risks that you're not going to be doing them at home. So a great example of that would be transcranial magnetic stimulation, or TMS. This is something that is FDA cleared for indications such as depression, but it is tested for things like chronic pain in an off-label way or electroconvulsive therapy. But again, this is something that you will need to be traveling to the doctor's office for each treatment. So you can see that might be a little bit inconvenient. And so what I really want to focus on is these home-based technologies. So these are technologies that are often powered by a 9-volt battery. They're very light. They're often wearable, or they're small, and they're handheld. And here are 3 examples of them. Transcranial Direct Current Stimulation, or tDCS, external trigeminal nerve stimulation, e-TNS, or TENS, Transcutaneous electrical nerve stimulation. And again, the names of the devices actually tell you a lot about how they interface with the nervous system. So let's think about that. I'm listing here the parts of the nervous system, the parts of the brain that these technologies are nominally supposed to be stimulating. So let's first start with transcranial direct current stimulation. Now, as the name implies, transcranial, this is through the cranium. So it's an external device applying electricity. The current passes through the scalp, through the skull, before reaching the brain. And so targets might be the motor cortex, the sensory cortex. So that's why I'm listing cortical targets here. All right, external trigeminal nerve stimulation. So the trigeminal nerve includes branches right here on the forehead. So you could put a device right onto the forehead, apply energy to the forehead, and therefore stimulate the trigeminal nerve. So this would be an example of a cranial nerve stimulation. Cranial nerves are a specific class of nerves that emanate directly from the brain, but can be accessed from the surface of the body, not just the trigeminal nerve, but other cranial nerves, such as vestibular for vestibular stimulation, or vagus for vagus nerve stimulation, whether it's the cervical branch or whether it's the auricular branch. And finally, you have something like TENS, transcutaneous electrical nerve stimulation. So transcutaneous meaning through the skin, really anywhere on the body where you have your peripheral nerves. Energy is applied to that part of the body, activating those peripheral nerves. And there are examples of particular peripheral nerves that might be targeted in the context of the management of chronic pain and migraine. So once again, we're seeing that there's different classes of technology, they have different names, and they're distinguished by which parts of the nervous system that they influence. And so the focus of my talk today is to try to explain in very simple terms, not too technical, how computational modeling, computer simulations, can be used to inform the design of these technologies, also try to explain how they work. So let's start with transcranial direct current stimulation, or tDCS, and present a computational model of tDCS. So this is what we're going to be talking about. Every computational model has these components. You can see here, this is actually a representation of my head based on an MRI. You can see that these are the tDCS electrodes. There's always two electrodes. In this case, one is over the motor cortex and one is over my forehead. Here's a cross section going through my body, and there's the nominal target. Okay, this is the motor cortex, and so this montage here is the motor cortex targeting version of tDCS, also called the M1SO montage for motor strip supraorbital region. Okay, now without getting into more technical details than we need to, all computational models, including the one I'm showing you here for tDCS, have just three basic steps. The first one is to represent the anatomy that you're stimulating. And so in this case, we're stimulating my head, and so we have to represent my skull, my scalp, and the brain. The second piece of it is you need to represent the stimulation device. And by that I specifically mean the electrodes that are passing energy into the body. And then just like in the actual device where we would apply current into the device and into the body, in the computational model, we represent applying current from the device through the electrodes into the body. And so essentially the model is simulating the application of energy into the body in the same way that it would be done in the real world. And finally, the final step is once we do that, we visualize, we predict, we simulate where current flows through the body and through the nervous system. And the result ends up looking something like this. So the way these results tend to be represented is with these false color maps, where, for example, in this case, hotter colors indicate areas where there is more energy being delivered to a particular part of the nervous system, whereas colder colors, in this case black, indicate parts of the brain where no current is flowing. Now, if you're looking at this, you might say, well, I'm not really seeing any black. And that is because in this particular montage, current is actually flowing through almost the entire brain. So when you put an electrode here, which is relatively large, an electrode here that is relatively large, the result is in fact a very diffuse current flow pattern through the head with these little hot spots that occur here and there, though you'll notice that the hot spots aren't necessarily under any one of these electrodes. They tend to be right in between. So this was a result that we published in 2009. And while now this sort of concept is very well accepted, at the time it was actually a little bit controversial because people were using this montage with the idea that it was specifically stimulating the motor strip, and these models did not support it. Now, what I've showed you before was the representation of the surface of the head. Let me now, because we can do this in a computational model, chop this up into different slices so we can visualize what's going on inside the head. And once again, you're basically going to see that there's no black. There's current flow everywhere. And so what we're seeing is that tDCS doesn't just stimulate the surface of the brain, right? The current actually cuts in. It cuts all the way through. And so this M1SO tDCS montage is actually stimulating not just much of the superficial cortex, but deep inside the brain as well. Okay, so we then went ahead and asked, well, where exactly deep in the brain is it going? And so in this later publication, what we did is we focused on specific nuclei deep inside the brain that happen to be known to be very relevant for pain processing. And in short, what we found was, it's not a surprise given that current was traveling deep, that the current was also reaching all these deep nuclei that have a very important role in the processing of pain, in the manifestation of pain. And so what we're saying is that this M1 montage, a montage that nominally was set up to target the motor cortex, in fact is stimulating much of the cortex, many regions inside the cortex, as well as many deep brain regions that we know are involved in the manifestation of pain. And so this leads to the idea that maybe a technology like this, tDCS, doesn't just work by stimulating one particular part of the brain, but it's actually stimulating multiple parts of the brain. In this case, we were thinking it's stimulating, in fact, the entire pain network. Okay, and so what we saw here is that we used computational modeling and we started to address transcranial direct current stimulation, and we asked whether tDCS really does just stimulate one part of the cortex, such as the motor cortex, and we saw that that's really not the case. It's stimulating subcortical regions as well, and it would probably be more appropriate to think of this form of tDCS not as stimulating one particular node in the brain, but rather stimulating an entire network. And so to understand any effects of tDCS on pain as a form of network modulation. Okay, now, having simulated where the current goes, I think we can now draw our attention a little bit more to, well, but what does the current actually do to the body, right? Why is electricity flowing through the body, in fact, leading to a reduction in pain, right? Why does zapping the body reduce headache? Why does zapping the body reduce back pain? And I like to suggest that, there's a lot of sophisticated approaches you can do, a lot of things like functional imaging to kind of assess this question, but maybe the most basic thing we can do is just ask people what they're feeling and look to see what it does to their body. And I'm going to give you very specific examples of that. All right, so let's think about TENS, or transcutaneous electrical nerve stimulation, where the primary target are these peripheral nerves. The main theory about how TENS works is something called the gate control theory of pain, or paresthesia. And this is something I've covered in other talks in quite a bit, but the basic notion is very much, is simply, if you rub it, it will feel better, right? If you bang your elbow, or your nephew bangs their elbow, what do? You rub it a little bit. The notion is that when you have painful stimuli coming in, and then you add on top of it non-painful stimuli, the non-painful input masks the pain, right? So that's the basic idea. And so the notion with how TENS works now is, instead of rubbing the arm, we put the TENS unit over the arm, we turn on the TENS unit, and the electricity that's pulsating into the body now starts to produce a tingling sensation because it's activating the peripheral nervous system. So the electricity pulsing in produces a tingling sensation. The technical word for that will be paresthesia. So that's the equivalent of the device. If you rub it, it will feel better. All right, so that's the mechanism. And it also explains why with TENS, we put the device over the part of the body that hurts. So particularly for TENS, right, this wasn't necessarily the case for these other technologies. With TENS, based on the fact that we think it affects the peripheral nervous system, and we have this gate control theory, we would put the device over whichever part of the body happens to hurt. Well, that's all great. But what's interesting is if you look at these other techniques, tDCS, e-TNS, in fact, all forms of non-invasive brain stimulation, the primary side effect that is reported in these cases is also tingling. People will report tingling, prickling, and so on. And so when you're using these other techniques, such as tDCS or e-TNS, which are not necessarily intended to affect the peripheral nervous system, you nonetheless produce, well, they're not intended to produce to stimulate the peripheral nervous system, but they are stimulating the peripheral nervous system. And I know that because I can feel it. All right, well, if I'm doing something like tDCS, and I think tDCS is supposed to affect the brain, but actually, because the current has to flow through the scalp, I'm producing tingling, how can I rule out that tDCS is actually not working by stimulating the peripheral nervous system, and therefore through gate control? You see, and by the way, it's not necessarily a problem from a therapeutic perspective, if that's what treats the pain, wonderful, but it is an important consideration from the perspective of trying to understand which of these are the actual targets, the nervous system targets of stimulation. Okay, and so this is really the question, and once again, I'm going to suggest that computational modeling is the way to address that question, right? And to try to disambiguate which of these are mapping to these techniques. All right. It gets it, by the way, even more complicated than that, because tDCS, I mentioned, has one electrode here and one electrode here. Well, that means that the current here is actually going through the forehead, which means the current here could be stimulating the trigeminal nerve, it could be stimulating other cranial nerves. So in fact, we see an interaction going all ways. How do we disambiguate that? How do we know that tDCS is actually stimulating this subcranial pain network versus just stimulating cranial nerves or just stimulating peripheral nerves? So computational models are a tool that will help address that with an additional step. I now want to use the computational models, maybe not simply to understand what's going on with an existing approach, but perhaps to try to make the approach better and more specific. And let me show you what I mean. So this is what I showed you earlier. This is the M1 SO montage, and this is showing you how current is flowing through the entire brain with hotspots all over. And so what we can tell from this approach is that we have two large electrodes placed across the head, and as a result of the fact that we have two large electrodes, we're going to have diffuse current flow everywhere. Working with colleagues like Alex De Silva, we thought, well, maybe there's another way to do things, another way to position electrodes on the head. And the hypothesis that we developed was, instead of using two large electrodes, let's use smaller electrodes. And let's position them specifically across the motor cortex. So what you're seeing here is one approach, where this is a bilateral approach now. There are two electrodes on one side, and then there are two electrodes on the other side. Or in this case, this is called high definition electrodes. There are four electrodes here in close proximity, and then the other side, four electrodes here in close proximity. Now, computational models tell us that with either one of these approaches, we are now able to deliver current focally to both motor cortex, to both motor cortices. I could only turn on one and just stimulate one side, but in this case, the approach was to do it bilaterally. So I want you to contrast that, of course, with this approach, where we're not delivering current necessarily specifically to the motor cortex with this approach here. Now, the other advantage of this approach is very little current slips to other parts of the body. So not only are we more focused as far as the motor cortex, but we also are avoiding things like stimulating the skin on the forehead, and therefore this confound of trigeminal nerve stimulation. And so what you're seeing here is that we're using computational models to develop new montages, and we hope that these new montages, for one, may produce unique benefits in the management of pain, and De Silva has shown how potent this montage is for treating things like chronic migraine. But in addition to that, by switching to these other montages, we now have the ability to more specifically say what was actually stimulated. So what I'm showing you here is this broad principle that we can use computational models to design montages that are more intentional to targets. Okay. And so we have a situation here where I had different forms of technology, and I said that there was this sort of muddling with some montages where you're not always sure what you're getting, and that using dose-optimized computational modeling can help you disambiguate what is going on. All right, so changing gears a little bit, I want to talk about e-TNS, or external trigeminal nerve stimulation, where really here the primary target is the cranial nerve, as the name again tells you, the trigeminal nerve. And I want to talk about a variation of this technique which stimulates the auricular branch of the vagus nerve. All right, so there's a primary vagus nerve running down the neck, and there's a branch of it that goes to the ear. And there has been a lot of work on putting devices on the ear, electrodes on the ear in very specific ways, in order to try to activate the auricular branch of the vagus nerve. But of course, you can put the electrodes here, or here, or here, or across So even when you have one target, there's actually a lot of variations of how you might try to activate that target. And so what we did, again, to try to, on our approach, is to try to use computational models and look at dose optimization as a way to try to understand what is the difference between these different montages and which might be more effective. As I illustrated before, the process, even though now we've gone from tDCS to stimulating the ear, the process remains the same. We need to represent the anatomy that we're interested in. So in this case, this is this is a subject's ear. And here we have sort of specific regions of interest. And these regions of interest are segregated by the fact that they have different cranial nerves or peripheral nerves going into them. So it does matter. This is not the same as this, is not the same as this, as far as where the current goes. And then the second piece of all these computational models was to represent the device itself. And as here, as I mentioned, you can see that there are different devices that deliver current to different parts of the ear. So here you have two electrodes here. Here you have two electrodes here. And so because the electrodes are in a different place, they will produce current in different parts of the ear. So even though it's one ear, we can have different outcomes. And again, I don't want to go through all the results in detail. I'm putting papers for people who want to dig more in, but not surprisingly, depending on where you put these electrodes, you will produce current to very different parts of the ear. And because the current flows to different parts of the ear, you will activate different ROIs. And so what I'm trying to show you here at a very high level is how computational models can be used to both tell you what you might be activating with your device, but also inform how you might optimize or alter your device for that purpose. And so we have here, again, the example of cranial nerve stimulation. And I gave an example of how we can use computational models with dose optimization, thinking about the auricular branch of the vagus nerve. Some colleagues of mine specifically looked also at e-TNS. And so here you can see just a snapshot of results from that publication. Again, at a very high level, they did the same thing. They had to first start with an anatomical representation of the head and of the nerve target itself. They started with a representation of the electrodes that were being used. And then third step, they predicted where current was flowing through the body. And as I suggested, they didn't just do one dose, they looked at dose optimization. So for example, in this figure here, they're considering the question of, well, you could have the electrodes here, or you can make a slightly smaller device and bring the electrodes closer together. What would be the consequence of that as far as where the current goes and what the activation pattern would be itself? And again, my focus here is not to get into the very specifics of, one target versus another target, one montage versus another, but just to explain to you in a clear way why computational models of devices and of dose variations can help us understand how these techniques work. Okay, so thinking again about mechanisms of action across all these techniques, I mentioned this sort of approach of like a very naive approach of, so let's just ask and look about how these different technologies work. And I previously mentioned tingling being a very common effect of all these techniques. Now, other than tingling, the most common reported side effect is skin redness. So whether any of these techniques, if you apply them to the skin, to the surface of the body, and so current is going through the skin, after you remove the devices, you will notice that the skin is very red, and the technical word for that would be erythema. And so this is normally considered a side effect of stimulation. It's not a serious side effect. The redness goes away by itself after about half an hour, but it's a very profound reddening. And it pushed us to start to ask the question, what if the reddening isn't a side effect? What if the reddening, which means this intense activation of the body's blood flow, is actually a mechanism of action? So this is what you're seeing here. This is a subject who received, in this case, tDCS. And so in this case, the electrodes were put across their forehead. after stimulation, the electrodes were removed, and there is so much redness, right? There is so much redness, you can see it by eye. So it's not a little effect. It's a profound effect in the body. Could boosting blood flow explain any of the therapeutic mechanisms of these techniques? Could, instead of thinking about the nervous system targets of stimulation, could we start thinking about the vascular, the blood flow targets of stimulation? So this is something we've thought about for a while, and others as well. We've called it neurovascular modulation. So adding that term vascular into the neuromodulation, which basically says maybe we're stimulating the vascular system, which is why we're seeing so much redness. Now, the notion that the body's nervous system's blood supply is important and in fact directly interacts with neurons is a very, very well-established technique, very well-established idea. This has been studied for decades by many people. People often describe the neurovascular unit, right, as a complicated interaction between blood flow and glia and neurons and interneurons, and it's really impossible to have a comprehensive view of how the brain works without considering neurovascular coupling. So that's really the starting point. There's extensive literature that brain function depends on neurovascular function. There's also a lot of literature pointing that a lot of diseases, possibly different manifestations of pain, different neurodegenerative disorders, dementias, are actually associated with a dysfunction of neurovascular coupling. So that's one piece of it. So we know that neurovascular coupling is important. The second piece of it is everyone already knows that neurovascular coupling is affected by brain stimulation. And the reason we know that is there are many techniques that are used to measure or image how the brain responds to stimulation that are vascular based. Perhaps one of the most well-known ones is something called functional magnetic resonance imaging, fMRI. fMRI is a technique that doesn't measure neurons directly. It measures the vascular response to neurons changing. And so there's been a lot of work using fMRI and other vascular-based imaging techniques to measure what neuromodulation does to the brain. And here's the thing, in all these studies, when they see a change in vascular function, they're not interested in it. They're assuming that the brain stimulation or neuromodulation is stimulating the neurons. The neurons, through neurovascular coupling, are then causing a change in blood flow, which they're measuring, but they don't really care about that. It's like a epiphenomenon, right? The fact that there's blood flow is an epiphenomenon. All right. So there's all this data showing that neurovascular coupling is critical for brain function. There is, hundreds of papers showing that different forms of brain stimulation actually change vascular function and neurovascular coupling. So we're just putting one piece on top of it, and we're saying, hold on a second. Maybe the brain stimulation isn't directly stimulating neurons, which is then producing a change in blood flow, but rather is directly stimulating blood flow, which is then producing a change in neurons. And so that's neurovascular modulation. And once you open your idea up, you open your mind up to this idea, it really opens up a spectrum of potential mechanisms. Because we're not just talking about one target, but multiple targets. Small vessels, large vessels, muscle cells that are involved specifically in the regulation of vessel dilation. There are also actually many parts of the nervous system, certain nerves that are primarily responsible for regulating what blood vessels do. The blood vessels themselves are not sort of one thing. You have sort of like from the smallest capillaries to the largest arteries to these peel vessels that then dive into the brain. All these represent potential targets for neurovascular modulation. And so when we're talking now about neurovascular modulation, it's really a exciting constellation of targets and potential mechanisms, including mechanisms that in fact impact brain clearance, and I'll talk a little bit more about that. And so I think from a therapeutic mechanism pathway, neurovascular modulation is very compelling. Okay, so I've talked about these different forms of brain stimulation, and I've talked about now that, okay, there's sort of these, instead of saying, let's say, neural targets, these system level targets. And so computational models help us guide us as far as which of these targets we might be going after. And then I talked a little bit about mechanisms, not just talking about neurons, but vascular as well. And it turns out that glia and other forms of cell are all part of this mix as well. Now, you can't just activate one of these cell types. As already explained, if you activate neurons, you will also activate a vascular response to neurovascular coupling, and glia will also be implicated. There's no situation in the intact brain where you can stimulate just one of these cells and not impact the others. Now, this raises a problem because now it becomes a little bit hard to know which cell is actually responsive to brain stimulation. Because if you do directly stimulate neurons, you absolutely will see a glial vascular response. If you directly stimulate glia, as some people have looked at, you'll produce a neuron and vascular response. And if you stimulate the vascular system, these will respond as well. And so the point is that the question from the perspective of neuromodulation is not which of these will be activated. I'm going to propose that all of them will always be activated because that's how the brain works. But rather the question is, there's a two-part question. The first part of the question is, when we apply stimulation, which are we stimulating first? Which is directly activated, right? Is it vascular first and then everyone else? Or is it neuron first and then everyone else? And I think the second very interesting question is, given that we are activating all of them, which is important for driving therapeutic benefit? We tend to default to think that it must be the neurons, but it does not have to be the neurons. Maybe, in fact, we're producing a vascular plasticity, or we're impacting brain clearance, and that is what's driving benefit. Now, because these are so tightly coupled together, it's in fact very hard to address experimentally, because as I said, anything you do that affects one will also affect the other. So once again, when we're trying to disentangle this, what can we do? We can go back to computational modeling, and we can use this modeling in a way that you couldn't do experimentally to try to disentangle the sort of who came first. I'm just going to show you, I'm going to finish with one example of how we did that. And again, this is going back to direct stimulation of blood vessels. And as with all computational models, we had to start with a representation of the relevant structure. In this case, we modeled not just the entire macroscopic brain, but also microscopic blood vessels. The technology I'm going to be focusing on here is, once again, tDCS, though I'm going to be showing you this applies for any case. So tDCS is being applied to the surface of the head, and you get this current flow pattern. Now we zoom in to one tiny part of the brain where we have these blood vessels, and we ask the question, how much of that current is flowing through the blood vessels? Does it concentrate in the blood vessels? Does it diffuse when it crosses the blood vessels? And what we found and reported in this paper is, in fact, as the current flows through the brain, it effectively concentrates into the blood vessels. The blood is much more conductive than the surrounding brain. And for other sort of biophysical reasons, the intensities of, in this case, I want to be careful, electric field that we see across the blood vessels can be much, much higher than what we see brain-wide. So that is this sort of broad notion of current being concentrated across these blood vessels. And again, the details are here. But this was a very remarkable prediction as far as the models went. I mean, it wasn't remarkable as far as the assumptions that we put into this model. We just assumed that you have a brain, you have these blood vessels full of blood. The blood vessels themselves, the walls of them are made out of these cells called endothelial cells, forming a blood-brain barrier that is rather resistive. So there were some assumptions caked in, but they were rather reasonable assumptions. And then when we ran through the consequence of these assumptions, we found this massive concentration of current across the blood-brain barrier, so that this quantity of a measure of current concentration, which is electric field, was massively higher across blood vessels than it was in the brain overall. And so, of course, this is consistent with the notion that stimulation is directly stimulating the vascular system. But what is the implication of this? So let's assume we are stimulating the brain with these different technologies, and the current is being funneled into these vessels, therefore across the walls of these vessels or the blood-brain barrier. What is the consequence of that stimulation? And once again, here we went first to our modeling to try to predict what was going on. Again, I'm providing citations to all these papers, and my goal here is not to get through all the technical details. I just want to express that we developed models that didn't just represent the entire head, didn't just represent vessels, but actually represented these tiny, tiny gaps between the vessels, known as between the vessels that form the blood supply. And these are called tight junctions. And to make a long story short, what we did is we used these models to predict what is the consequence of all this current being funneled through the blood vessels. And our prediction is that this would drive brain clearance. So your brain is always trying to clear things, right? Your neurons are working, all the cells in your body are working. There are byproducts, metabolic byproducts of that activity, and the brain must somehow clear those through different mechanisms. And one of those mechanisms has to do with this sort of slow transport of water out of the brain. The details here are less important than the message I want to get across, which is that we use these computational models to develop a very specific theory for how, in this case, tDCS might affect the brain. And these computational models predicted that tDCS would actually boost brain clearance mechanisms by driving current and concentrating it across these vessels. Now, of course, these are just predictions. The predictions need to now be verified in different experimental ways, but it will always remain the case that there's certain things that are really hard to disentangle with doing experiments in people or even doing experiments in animals. And so what I'm trying to draw your attention to is the place where computational models sit, not just in trying to explain where current flows depending on where you put the devices, not just explaining how you should optimize where you put the devices in order to direct current to a specific region, but also to give us very unique insight into the mechanisms of brain stimulation, neuromodulation, whatever those mechanisms will be. And really, I'm just showing you one suggestion here. Okay, so I hope I've achieved my goal as just described. And certainly, if you want to dig in more, I have plenty of other videos on both the mechanisms of neuromodulation and also how neuromodulation works for pain.